All right, good morning, everyone. So my name is Tatiana, someone doesn't know me. So today we have Dr. Brett, and he's going to talk about the ecology of abortions. So we're going to see a little bit of disease and what vets do in the field. So Brett is graduated from the University of Pretoria from South Africa and has been working on different zoos for more than 15 years. You have a lot of work done in Africa and in the Middle East, Philippines, and in the United States. And he recently moved to Australia. It's only 7 years, so it's not that recent. He moved to Australia. So now you're completing your PhD at the University of Melbourne, and he's looking into the infections caused of abortions in Australian feeder seals, which you're going to show us a little bit today. And Brett is based part-time in the Werribee Open Range Zoo, one of the zoo's clinical veterinarians from the zoo, and the other part-time doing PhD. Outside this is ecology, Brett has especially interested in field of aquatic anesthesia, especially species at Epos and Phil. And we're going to see Epos pictures.
I can put one in.
Okay, cool.
Alive. The Locus are great stories, anesthetizing hippos.
Yeah.
Doesn't turn out that well for Max. Well, good morning. Thanks for having me here. I know that this is probably not something that you would normally discuss over tea time. Infectious causes of reproductive failure and abortion. As a heads up, there will be a lot of reference to miscarriage and abortion and neonatal death and pictures of dead things. So if that's not your cup of tea, you might be in the wrong lecture hall. But thanks for the privilege that I can chat to you today about reproductive failure in Australian fur seals and what we know about it. So I don't know how many of you in Queensland have actually even seen a seal. We don't have any pinnipeds at top here, but the Australian fur seal is one of those really omnipresent apex marine predators that historically during the colonial era of Australia was nearly wiped out in large portions of where it formerly used to breed. And they've come back incredibly well since the cessation of commercial harvest. And what's recently been reported, well, not that recently, but in roughly about 2010, 2011, is that about a third of them are losing their fetus in the third trimester. And in one of the largest breeding colonies, they've had a continuous increase in pup numbers. And over the last few years, it's progressively decreasing. And so the question is this multifactorial or nutritious? So we want to try and find out, is there any infectious cause that's responsible for this, because these are really important indicators of ocean health for us, being an apex predator that is a generalist using everything in that benthic space of the bass strait. So if you have a look there, there's a beautiful little stillborn, one of my favorite samples. This is an abortion. So if you have a look at that, it really doesn't matter in the end what it is. Embryos can die from really early on till right after birth. And all of that is going to sort of classify as reproductive loss. But for us to work on early embryonic death on remote offshore islands is almost impossible. So our entire focus is from when it looks like a seal pup and it's pink, and it looks like a seal pup and it's furred and it's born dead. Anything really early beyond that and really late after that, I won't be discussing today. But most of the causes that we're going to chat about today, which is that top infectious, one can affect reproduction right through until well after birth. So these are just some of the common causes of abortion in mammals. And in marine mammals, we really don't have a lot of them that we're aware of, a lot less than we have in terrestrial animals. So in marine mammals, we really have three things, especially in Australian fur seal breeding colonies, that are quite important. So nutrition always plays a role in wild animal populations. In Australian fur seals specifically, we're not exactly sure how much it plays a role compared to the Cape fur seals, which they're closely related to as subspecies of. Those are pelagic foraging. They've got really nutritious, high quality diet. Well, high quality, I should say, very high nutrient levels compared to the Aussie fur seals that are benthic foragers. So they're eating on the sea bottom and diet that is slightly less high in caloric density. Whether it's that important, we're not 100% sure. But infectious diseases is something that's being heavily neglected in them. There's a little bit of work on toxicities. So I don't know if any of you are aware of a thing called persistent organic pollutants, but a lot of us will have waterproof jackets, Teflon pans, a variety of things, and that's all created in the process of making those chemicals. And unfortunately, persistent organic pollutants, they persist, they accumulate in apex predators because they often are predisposed to bind with fat. So we have found a little bit of evidence of them, not in work that I've done, that one of our colleagues has done, that they're there. But the big thing with them is they have great synergism with infectious diseases and will often help infectious diseases to seed. Within Australia, we don't have any signs of harmful algal blooms, which is great because that is a really major cause of reproductive failure when it is present in marine mammals. So we're going to focus on that top one, but I just wanted to show you what a harmful algal bloom looks like when it hits a seal colony that's naive to us. This is in Namibia, so this is a caged fur seal breeding colony. There's over 1 million individuals in that area breeding, and this is drone footage, and every one of those tiny little black specks is a dead stillborn or an aborted fetus, and there we have over 20,000 pups produced in less than a week from a sudden change in the ecology, having a diatom soil called Phylogenetic. Not that that really matters to you, but it produces a really dangerous neurotoxin, which in humans is known as amnesic shellfish poisoning. But in seals, it actually makes it impossible for the mothers to navigate because it affects your sense of direction. And one of the other effects that it has is abortion storms. So we haven't seen this in Australia in fur seals, and luckily so, because there are more than enough other things to look for, because there's a lot of infectious diseases, and we know more about what happens on land because it's easier to study what happens on land. So if you go offshore into marine mammals, all of the seal type things, pinnipeds, are a lot easier to study than cetaceans, so whales and dolphins. And within them, we've got predominantly 4 infectious disease classes because harmful algal blooms are not considered infectious, they're considered toxic. And Tatiana here, almost everything we're going to look at can infect you as a human being as well. So it's called zoonotic when it can come from zoos, from animals to humans. So the more we look, the more we'll find. So we've chosen 2 of the biggest breeding colonies for the species. This is the Bass Strait down near Melbourne, which luckily none of you are there at the moment. It's pretty miserable at the moment. And these offshore islands are pretty cold and miserable places, but really beautiful if you're a seal for breeding on. We've got seal rocks over there, which this is just outside Phillip Island. That is the biggest breeding colony for the species. There's over 18,000 individuals breeding there, all producing their pups in roughly a month and a half. And then Kanana Island is the 3rd largest for the breeding colony for the species, and that one's just offshore from Wilson's Promontory National Park, so a lot more pristine. But all of this is within the Bass Strait, which is a really shallow oceanic system that changes very rapidly historically, and probably with anthropogenic influence is going to change progressively more. So that's Kanana Island, really beautiful, really pristine and offshore. This is seal rocks. So this is less than a kilometer from a really highly tourist visited beach and Western Port where there's a lot of human activity. And then in between these tightly packed sardines of adults, we're looking for these. And this is a 3rd trimester abortion. And the way we find them quite commonly is a gull. There's actually a silver gull busy wrecking the placenta on that sample. And that is our main focus, is trying to find these 3rd trimester abortions and find out why there's such a high incidence of it. So you can imagine going out there, it's not really easy to get diagnostic samples. If you consider in human miscarriages, the majority of them go undiagnosed as to actually what caused it. And that's a human being that's going for regular ultrasound, blood tests, etc. So you can imagine an offshore breeding colony like this, the ability for us to diagnose the cause of abortion is quite difficult. Compared to like well-monitored dairy herds in Europe, there you're getting 20 to 75% of abortions will be diagnosed as to what caused it. And the thing is that in most of these instances, it's an infectious agent interacting with something else. So we've got other things to take into consideration, these lovely gulls, both Pacifics and silver gulls that unfortunately can get to the seals quicker than I can. So a lot of our samples we don't get to. There's cod rush in and the seal stampede if you do so. It's a very lengthy process of crawling through seal excrement until we can get to the fetus and then crawl out with it in a bag. And then we'll do a variety of things, which I'm not going to go into because that's all Betty type stuff, but we look at it macroscopically, so with the naked eye, then we'll cut a ton of samples in and we'll fix them and stain them and look at them under the microscope, both just normally and with a variety of special stains to look for certain pathogens. And then what's really important nowadays is molecular testing, because a lot of these pathogens, if you cut the slice in the wrong spot, you don't see the bug. But if you do a molecular test, you can say it's there. So culture is a little bit more old school and a lot more difficult for a lot of the pathogens we're working with because they don't grow very well. And we'll go into that a bit later as to why they're so difficult to culture. But the main aim of this study really is actually just to see what is present in Australian fur seal colonies that we can say, is it there, is it not there? And if it is there, is it significantly associated with third trimester abortion? And the way that we're using the control for this is full-term placentas. So we can't really get any other control in that type of environment. So we're doing a statistical comparison between the two. And then based on that, our sampling is sort of set up that we can say, is there anything we're missing? Because we're being pretty thorough in the techniques that we use. So that's where you better try and get into. And this is full-term placentas that we're harvesting there from where the pups, when they get to a certain age, start crashing. So beautiful place to get samples from, but there are a few bulls hanging around that we have to try and dodge, normally protecting their harems. So within specifically Australian fur seals, not all marine mammals, not all pinnipeds, there's two bugs, one of them being a very important bacteria, Leptospira, that's water associated and commonly spread by rodents, quite common terrestrial cause of abortion. And morbillivirus, which is the same as distemper, which we get in dogs, it's in the same group. Those two, we can fairly safely say there's no evidence of them yet in Australian fur seals. So it's always easier to say it's there than to say it's not there. But we are very interested in this organism called Mycoplasma, because there is some evidence that it is present in these colonies. Brucella, because that is one of the most important causes of abortion in domestic animals terrestrially. Chlamydia. I don't know if any of you here work with koalas. Okay, all right. Well, you should be aware of this disease because it is the most important cause of infertility and reproductive disease in koalas, especially in Queensland, and a key contributing organism to their decline. It's also one of the most important causes of abortion in horses in Australia, where it has a synergism together with the herpes virus. So we wanted to look for that. We also want to look for this little beautiful organism there. That is a microscope slide of a little protozoa known as Toxoplasma in the heart fibers of an aborted seal fetus. And then this organism, which was actually diagnosed for the first time and described in the 1930s in Queensland. It's known as Q fever. So we're going to go into those four that we found, because chlamydia, there is no evidence in any marine mammal, both north and southern hemisphere at present, that it is a cause of abortion. Sorry, before I dry mouth on all of you. I'll put the slide up twice because I think it's really beautiful and it's very difficult to find this life stage.
What is it?
Right over here. So these are all little tiny little toxoplasmas waiting to get eaten by something else so that they can infect it. And toxoplasma is an intracellular protozoal parasite. And you're going to see on everything today, I'm going to say intracellular, because the placenta is this giant fortress that protects the fetus and is almost impenetrable unless you're a really conniving and clever pathogen. And all the conniving and clever pathogens that can get across the placenta tend to be intracellular. So Toxoplasma is very important in Australia and worldwide. It's spread by domestic cats. So I see you had a cat scarf on earlier. I'm assuming there's other people here that own cats, and it's your own little friendly cats that live in your garden, in your house, and some of the ferals that are unfortunately pooping throughout Australia, spreading this parasite. It is quite dangerous for marsupials because they have not co-evolved with this pathogen. And normally it is a cycle between mice and cats, because those two traditionally have co-occurred. It is transmitted horizontally and vertically. I don't know if any of you know, but horizontal is through the food chain, whereas vertical is it goes from the mom through the placenta to the fetus. So we've now got evidence that it does go vertically in southern hemisphere marine mammals, but most of the work actually has been done in northern hemisphere marine mammals on this, and it's been found to be very important, really important in sea otters. They're kind of like the textbook poster child of Toxoplasma. In them, it is a definitive cause of their population declining. It causes abortion. It also affects their nervous system, and it makes them more susceptible to great white predation. So what I've got up here is a purple sea urchin from Port Phillip Bay, but sea urchins are thought to be, they're hypothesized at the moment as one of the really important ways that sea otters are getting infected by grazing. These little guys graze on algae, and then the oocyst, which is the infective stage from the cat poop, is sitting in the algae, and they graze it, and the otter eats it. In birds, because they can't really abort, because the eggs are already out and about, it does tend to affect the spleen really heavily, and the liver, because birds, has anyone here ever looked at a bird cell under a microscope, a red blood cell? They actually have a nucleus compared to an animal. And this pathogen can only infect nucleated cells, so it jumps into the red blood cells of birds and therefore can infect other organs. But in Australian fur seals, we really, until recently, didn't know if it was significant. There is very little data about Toxoplasma in southern hemisphere marine mammals. There's a single case describing it in the New Zealand fur seal, found dead near, well, dying near Sydney, and then it's been described in 2 species of dolphins and in little penguins. There's a lot of it in terrestrial mammals in Australia, so we do know that it's here. But what's very significant for us is that we found it in 7.5% of all the aborted fetuses that we've collected from both colonies. And we haven't found it in any of the full-term placentas. So it does appear that it is quite significant. How are they getting it? I have no idea at the moment. So don't be cheap like me. Get BioRender, the paid version. It won't pixelate when you go onto a big screen. But we've got cats. We know they're in Australia. We know that they're pooping, and we know that there's rain in rivers, and that their oocysts are probably ending up in the water. This portion here, we have no idea. We do know that the Australian fur seals must be obtaining it through their diet, and we know that we have found aborted fetuses where it's been transmitted vertically. So that entire horizontal part is still missing. And that's something that we'll have to figure out over time. But we know that this is one of the most important pathogens that we've identified to thus far in Aussie fur seals. So we'll go over to our next suspect. The reason I chose this sickly looking pup for Mycoplasma is, if you have a look, it's got quite a runny nose. So Mycoplasmas are one of the smallest, weirdest, most difficult to work with little bacteria that originally was thought not to be pathogenic. But progressively, the more and more we are able to study them, the more we've realized that they are omnipresent in all species of mammals. And in a huge portion of those mammals, they are very significant contributors to allowing other pathogens to come in or creating pathology themselves. And they are typically associated with mucous membranes. So mucous membrane on the front end of the seals, nose and mouth, back end reproductive system. So these are just 4 of the species that have been identified to thus far. There are probably way more of them. And historically, most of these were actually associated with respiratory disease and bite wounds from seals. There is a wretched thing called seal finger. So if you work in marine mammals long enough and you eventually get cut or bitten by a seal, you are going to get a seal finger, and it is typically caused by these guys, and they're impossible to get rid of. But very recently, it appears that this pathogen is actually associated with abortion. And what we're thinking is that it is potentially almost like a gateway drug. Like, not everyone that smokes marijuana is going to do crack cocaine, but almost everyone that does crack cocaine started with marijuana. And Mycoplasma looks like it might be a gateway pathogen that actually allows other pathogens to come in by altering the immune system on mucous membranes. So if you look at this here, that bar on the left, that is in full-term abortions. And in those, we're seeing well over 50% of them that are positive for Mycoplasma. And if you go to the full-term placentas, which are presumed healthy controls, because we can't rule out that all of them are born are going to be healthy, but there we've got just over 20% where it's present. So it does seem that this pathogen might be quite significant. It is just really difficult to grow it and work with it. But going over to one of our other suspects, this was originally what we thought would be one of the most important pathogens. If you look at its name here, for the first one described, it is Brucella abortus. So it doesn't really give much to the imagination as what it does. They're very important pathogens in domestic animals and in a lot of terrestrial wildlife. They cause major amounts of abortion in some herds of goats in various parts of the world. And they are sort of a textbook case of a pathogen causing abortion. What's really bad about these guys is they often infect a lot of other things as well. And so humans get central nervous system infections from them, bone infections, another one of these pathogens that we want to be careful of not getting. You'll see that every one of our pictures I've got gloves and things because I want to live long, not long and painful. But Brucella itself in seals is identified as Brucella pinnipedialis. There's a lot of argument about how do we taxonomically group them. It doesn't really matter that much because what has been found to thus far is that there's probably two species with a lot of variety in between them. Seti, which infects cetaceans, so whales and dolphins, that is a super important one. But we're not talking about whales and dolphins today. We're talking about odriids, eared seals. And eared seals, it appears that pinnipedialis doesn't really play that important a role in them. So this is a placenta. I know there's a lot of microscope pictures, but at least it's not the dead thing that you can see. It's a beautiful picture of it. But this is a huge amount of inflammatory cells infiltrating a placenta, causing it to dehisce and pull loose from the mother. So what we've seen with Brucella is that there's actually not a very large portion of these fetuses that are positive for it. And just because it's there doesn't always mean it's causing pathology. And unfortunately, in a large portion of these where they're positive for the organism, we're not actually seeing placentitis. So this typical lesion. So this suspect is probably lower down on the list of importance, but this is where we now need like really good music for this big bad guy coming in. Like one of the most important pathogens is this one. So Coxiella burnetii, something that we've never really ever surveyed for, but probably one of the most important causes of abortion terrestrially. And considering that Australia is one of the developed nations in the world with the highest prevalence of it, something that we should have looked for earlier. It's never been diagnosed before in the Southern Hemisphere in marine mammal. But it used to be super common in abattoir workers, but progressively, the more and more we're surveying people working in wildlife. So whether that's researchers, wildlife rehabilitators, and so on, a lot of them have got antibodies against this, showing that people that are working outside of the abattoir industry are getting exposed to it. And the original belief is that this intracellular bacteria in marine mammals was of terrestrial origin. So I'm assuming none of you even know what you're looking at. But this is a placenta, and these beautiful little clusters of grapes, those are huge, sort of jam-packed cells full of Coxiella, where they're actually destroying the entire placental structure, and that's what causes it to loosen and for the abortion to occur. So it's been identified in 5 northern hemisphere marine mammals. In 2 of those, it is associated with species that are showing significant population decline in populations where they actually have this pathogen present. So yeah, it's not really that easy. We can't throw 100 seals into a thing and infect them and check if they're going to abort, but suspicion is really there that this organism is probably causing or contributing to those abortions, and that is specifically in stellar sea lions and northern fur seals. I just put this in here for you, Tatiana. It's not for the rest because no one's going to care about that. Those are 3 PCR markers. They're molecular little segments that are present somewhere in your genome, and we use it to find them. They've different uses, but that one there, IS 1111, is called the gold standard. So if you went in and you've been out and you've worked in a seal colony, so if Kate came back and she's not feeling really great and you've worked in the seal colony midsummer, that would be the first one that you'd normally be tested with. It appears that it is actually absent in marine mammals. So that is very weird because it shouldn't be that way for Coxiella. That is considered essential for a Coxiella to be Coxiella. So after all the work that we've been doing with Coxiella, we were fortunate enough to grow some of it, and we decided we're going to sequence it because a lot of our stuff is thumbsuck extrapolation if we don't have the genome of this organism. So we want to know these little beautiful little dead seal bugs there. How is this causing that? And one of the most important things, which I'll go into later, is this MLVA. And I'll explain that a little bit later, but it's an identification tool that will allow you to take bacteria and look at them. If it's the exact same bacteria, same species, are they from different geographical regions? And have they got a temporal scale that splits them? So it's a very important tool. The other thing that we do is taxonomy, which I'm not great into. People help me with that, but that we can just see, is it actually Coxiella? Does it relate to everything else? And then what's really important is this thing actually causing pathology? And in Coxiella, there's a lot of things which we call virulence factors, which if you're a big gamer, and you're into like these quest type things and you get skills and add them onto you, virulence factors are like adding these special skills to you that make you more dangerous. So IS11, don't worry about how we found it. The only thing is that it isn't present in the genome. And that fits with all the diagnostic testing that's been done in the Northern Hemisphere where IS11 doesn't amplify in the samples that they've got from those animals. It also fits why we couldn't originally find it with that. So we're using 2 other PCR markers now that we know it's not present in the genome. And then just call it MLVA, but I've written it out there for you that you've at least got it. MLVA is like a little fingerprint, like I said, to distinguish bacteria based on their geographical and temporal scales. And we look at multiple repeating little small alleles within the genome and then look at their size and where they're present. So if you have a look there on terrestrial Australia, we've got 3 loci, MS24, 28 and 33. That is super specific to Coxiella in Australia of terrestrial origin. It is not shared with other parts of the world, so it allows us to pick up where an animal has gotten infected. What was very interesting is that when you look at the bottom there in black over the ocean, that is the MLVA sequence from Australian fur seals. And there is only one locus, MS28, that is shared between the 2. So it is very unlikely that in recent evolutionary terms, this is a terrestrial spillover. So how did it get into Australian fur seals? We don't really know. Is this a migration from northern to southern hemisphere, or is it from west to east? But it's very likely that it hasn't come from the land into the water, even though we've got huge amounts of secretion from goat herds up in Queensland, goat herds down in Victoria, macropods that can carry it, but it looks like it's its own thing in the water. And what we then did is just wanted to see, is it actually Coxiella? And I'm not going to go into this. It clusters really far away. It is still Coxiella, but it is a very distant, it's like your relative that you almost never see, but you do have some relation. But when it clusters that far away, you want to make sure that you're not misidentifying the data on the genome because you can interpret it wrong. So we looked at a thing called housekeeping genes, and there are certain genes that are preserved in each bacteria that bacteria needs to remain that bacteria. And if you look at those, Coxiella burnetii, that PG457 is just its naming code. It is together with all the other Coxiellas. These other 2 bugs there, one of them is a Coxiella in ticks, and most of these are actually what we call endosymbionts. They're non-pathogenic Coxiella-like organisms that live inside ticks and have co-evolved together, and they don't cause pathology to the ticks. So one of the hypotheses is that Coxiella burnetii is an recent evolutionary organism that's come off of an endosymbiotic bacteria. And if we then go into the really beautiful part of it, which is the superpowers of this bacteria to get across the placental fortress and cause abortion or premature birth, is these 2 things. Don't really worry about what they do, but lipopolysaccharide and type 4 secretory system are these incredible tools that allow this organism to pass through using host cells and infect the placenta. And because of that, we went and looked for them. These are just the code names for what they're called on the gene. This is, are they there or are they not there? And that is, how much percentage similarity is there to the highly pathogenic Coxiella that we know is going to cause disease. And so if you can look at that, the top 6 are all critical to the type 4 security system and to the lipopolysaccharide. And yes, they're there, and there's a very high percentage of similarity. So that means we probably should, based on a sort of analysis of in silico work, consider this a pathogenic bacteria, and that is probably quite dangerous, but we can't really go out and just randomly sample seals everywhere throughout Australia and catch them. It does cause a lot of chaos. So what we've started doing is collecting environmental DNA. And I don't know if any of you use eDNA to survey for species in the environment, take a water sample and tell you if your fish or whatever you're looking for is there. But everywhere we walk, as I'm walking here now, I'm shedding skin. And in a few days from now, you can swap the carpet and you can say that I was here. And so what we do is we collect soil from these breeding colonies and then look to see if Coxiella is there. And this is during the period immediately after pupping. So all of these seals are going to pup sort of over a month, month and a half, December, January. And if you have a look here, the red dots correlate to how positive or negative the sample is. So the bigger, the better, and the blue dots are negative. All the blue dots tend to be either on the outskirts of breeding colonies or at elevation. And if you look at these, it means that certain areas, which are the prime areas where all the large females congregate, are the low-lying areas where the big fat bulls hold in harem. All the younger females that can't compete as much for a mate tend to go to the higher areas. So is there a difference between the different groups? I don't really know, but what we do know is when we go immediately after the breeding season, in the middle of winter, when it's pouring with rain in Victoria, all the environmental DNA washes away. And if you have a look here, there's only one little remnant sample in a tiny cave where they pup. So it appears as a very sensitive tool to go out to the environment and check for things. It's something that's not limited to disease. So you can use it as an ecological tool that's really handy looking for endangered species that are difficult to monitor. And it's very handy. But as I said, we often use gulls to find them because when you're sitting above a few thousand seals trying to find an abortion, our eyes are not great and I can't fly a drone constantly. But these guys are drones. The only thing is that they're also competing with us for the resource that we're trying to test. And that is a Pacific gull. If you look at that beak, other than it's a pretty beautiful bird, but that is a fat beak with a really big build depth. And they've got the biggest build depth of any of the gulls, as far as I understand. So this is Kanowna Island. Seal Rocks is over there. This is roughly about 40 kilometers as the crow flies or as the gull flies. It's called Seal Island. There are no seals. There historically used to be seals. They got decimated. They haven't come back. But we've got a population of Pacific gulls that breed on Seal Island, and we've got a Pacific gull colony breeding with Kanowna Island. And this is really cool resource study because the bunch on Kanowna during the breeding season of the seals forage almost exclusively on placenta. They actually just gorge themselves on these placentas, whereas the bunch on Seal Island do very, very foraging for other types of resources. So they're a beautiful control for each other to see what role do gulls play in transmitting Coxiella as a vector through the environment. So Mer said that IS 1111, we've thrown it out. This COM1 and HTPAB, they're just 2 PCR markers, but they're really good PCR markers for Coxiella. And I've made the 2 islands there, red for the one with the seals, yellow for the one without the seals. And oral just means we swap their mouth, cloacal, we swap their cloaca, and when we took blood, and in theory, if you're a mechanical vector, the bacteria should never be in blood. But if you develop a response and disease to it, potentially you could become bacteremic, so the bacteria will circulate through your blood. And if you have a look, on blood, we couldn't detect it in Seal Island. On blood, cloaca, and oral, on both PCR markers, it's very common for the gulls to get it. So it's pretty likely that they're playing quite an important vector role, whether that's purely mechanical or if they're actually amplifying or transmitting it by going through their blood, I don't know. But what's very interesting is that if you look at the Seal Island group, there is still a very low level of positives. And that means that, so there must be occasionally some of these gulls that are coming across and foraging where the other gulls are protecting territory. The important thing about this is that gulls want fresh water. And these islands do not have fresh water. So what could happen with these birds, and this is some tracking work that we're going to be doing with them, is to see when they fly out from these islands back to the mainland, where do they go? Do they come to popular beaches, good swimming spots, dairy farms? And could they potentially be acting as a vector for spreading this pathogen back onto the mainland again? We don't know. What we don't know either is whether or not Coxiella, because these animals are coming back to remake, getting it in their bloodstream, is it going to affect the viability of the chick hatch? So we have no idea. We've never compared successful chick hatching between the two colonies, and we don't know if it's causing anything. But what we do know is that they are incredibly important in the ecology of Coxiella. Exactly how, we'll still have to find out a lot more, but that is what a typical breeding season looks like. There are just gulls flinging placenta everywhere. And during the non-breeding season, when there's an abortion, you can barely get to the fetus because you are competing with every single gull going for that one limited one. So, as you can see, there's a lot of pathogens out there. And I know it's not something that you'd normally discuss in ecology. A lot of your stuff is going to be telemetry, resource. These are invisible organisms that are intricately involved in ecology. They control populations. And if we don't know the baseline, whenever that baseline alters and we get massive die-offs and suddenly there's a pathogen present, there's always the question of, was that pathogen always present? Is it a novel pathogen that's being introduced like Toxoplasma? So it's really important in any species where you're trying to understand the success or the failure of that species to thrive, what the baseline of infectious background in it is. So this is purely on a reproductive perspective that we're looking at it, but a lot of these pathogens will have non-reproductive effects as well. So I just wanted to thank a large amount of people. I'm not going to name them all, but I'm definitely going to name my supervisor, Jasmine, because when I started this PhD, I was a very clinical vet that specialized in anesthesia. And she's allowed me to actually learn how to do research properly and think more as a research veterinarian and as an epidemiologist rather than an individual case-based researcher. So if you ever want anything, reach out to me. My contact details are on there. I love disease. I love disease epidemiology. I also love non-sick animals. And you can learn a lot from that's a non-sick seal. We're just tracking it. There's nothing wrong with it. But yeah, please feel free to reach out. And yeah, if anybody's got any questions, see, there was going to be a nice, healthy looking seal, not dead, not dying. Technical stuff.
I do have one question. Can I start with the questions? So for those, so Coxiella bonaidae cause Q fever, and in Australia, 1 in 20 Australians have been exposed to Q fever already. So it's a very important disease. Not many people know about it, but it can be very dangerous. So I actually know 2 people who were hospitalized last year because Q fever. Is this Q fever the same Q fever, the Coxiella? Can be zoonotic, this one? Do you think that because it doesn't have the gene?
No, so we actually, so that one missing sequence, that insertion sequence doesn't, as far as we know, have anything to do with its pathology. If you look at the virulence factors, so those things that code for lipopolysaccharide and type 4 secretory system, they are definitely present, and they are present in a way that they are very similar to highly pathogenic Coxiella. If you look at a lot of the people that have historically worked with seals back in the day, irresponsible days, no PPE, a lot of them have had recurring fevers.
Okay, yeah.
And one of the things that you get with Coxiella is recurring flu-like fevers that come and go that respond to doxycycline. So there is a fair likelihood that it is able to do that. Can it cause abortion storms like we see in goats? We don't know. Can it cause fatal heart disease in humans? We don't know. But about 5%, I think, you maybe correct me on this because I'm not that good at it, but I think roughly 5% of human beings that get clinical Coxiella can actually develop cardiac disease from it, and that cardiac disease can be fatal. So what we have started doing is actually recommending vaccination for people that are working in first seal colonies.
Yeah, so yeah, Australia is lucky that we have a vaccine. No one else has a vaccine.
Yeah. Yeah, and then I'll do best.
Yeah, great. Thank you very much. Just for remembering all those long names. Incredible.
No, well, if you were talking ecological jargon, I would also be going, wow.
Anyway, any solutions? So especially for the one that's kind of human induced, and can any ideas of mitigation that can be done or should be
It's
done?
an exceptionally good question, and that's why you need to know what is there and what's not there. And so Toxoplasma, we know with fair certainty can say is an introduced disease within Australia because cats were never present here. So, and it's not something that the only host that can act as a final host. So a final host is a host in which a parasite can have sexual reproduction. And so if you do not have domestic and feral cats free roaming, you can automatically decrease and eventually probably mostly eliminate the delivery of oocytes into the environment. So we know that domestic and feral cats are really important for predation of native wildlife, but they're also very important from a disease perspective. And people that are naive, that don't own cats, so women that have never been pregnant, that have never been exposed to Toxoplasma, for them it also has a reproductive risk. So in those type of instances, you could go, if we became more stringent on cat control, that 7.5% of reproductive loss, we could possibly alter that and have massive other benefits to wildlife as well. Things like Coxiella, we can actually vaccinate for in humans, but vaccination in wildlife is still very hot topic, touch and go, whether we should or shouldn't. But there are on some things interventions. Mycoplasma is very difficult to do anything for, but Brucella one can also vaccinate for. So once one actually knows what is the baseline, there are some of the organisms that we could probably intervene on, some of them on a management of the source, and on some where a vaccination type protocol might be important, but then that's an entire different ethical debate. And then also some of these vaccines, if you actually have been exposed and you don't know it, you get the vaccine, it's not really good for you because you can have a horrible reaction to the vaccine. So that'll be another discussion. But there are definitely solutions if one's willing to go the route of that. Steve, sorry.
So since you've seen an increase in abortion in the first seals, and the goals feed on the placenta and stuff, has anyone noticed an increase in the gull populations because of those increased abortions?
So just to quickly correct, so there's no increase in abortion because no one's ever done proper full-on study what is the abortion rate and the change over time. But what has been noticed is that there's a disproportionately high level of abortion in the third trimester, and marine mammals have a higher propensity for aborting in the third trimester because lactation is so intense for them. So compared to other species, it's quite high, but it's only a single study that detected.
I see. So there's no like that.
There's not long-term data on abortion rates, but what there is long-term data on is from the biggest breeding colony, which is Seal Rocks, that there was pup increase, stabilization, and now progressive pup decrease. Whether or not that is from abortion, failure to conceive, environmental, because Seal Rocks is quite a low-lying colony, so with severe weather events, there's chance that we're losing pups from storm washout or so on. So, so very difficult to say, is that pup decrease definitely because of reproductive failure, or is it multiple factors playing together? Is there an increase in gull populations? That I actually don't know. There might be data around it, but there's a lot of data that there are huge incursions of them inland, and there are some areas where there are proposals for gull culling because of the large numbers. Is that a result of anything together with the seals? I have no idea.
It could be an interesting to compare, see if there's long-term data on those seagulls. You know, you could maybe like give me one reason as to why they increased if they have increased over time.
So it's a very good question, but unfortunately one that I have no answers to really.
So if a female, if a female is infected, does it can carry that infection its whole life, or is it all like de novo infection?
So that really depends on the pathogen. Okay. So each pathogen has a different route. And some of them will remain, like a Mycoplasma. If you look at human beings with recurrent spontaneous abortion, that mycoplasma or ureaplasma, depending on which species, is involved every single time. Some other pathogens, there's the potential that if you are naive and you get exposed, you will lose your pregnancy, but then you develop immunity over time and the chance is less. So it depends on which pathogen you're looking at. Toxoplasma is one of the pathogens where there's a suspicion that over time maternal immunity develops and the risk of abortion decreases. But in a lot of these colonies, we don't know because we'd have to do major extrapolation from what's known in domestics.
So possibly the age structure of the colony might have quite an influence on reproductive loss.
And then also, if you're the older, bigger females tend to be able to hold those better areas. So for certain pathogens, they might have higher exposure, but a better chance of a mate. We don't know in a lot of these diseases what the male influence is, because some of these pathogens, the male could also be an amplifier. So there's definitely going to be population differences. In that paper where they did this, where they recorded that there's a higher or a disproportionate number of third trimester abortions or pregnancy losses, there was a suspicion that there was an age class difference in the females. So that would allude to that potentially the factors are not the same in all age classes. But yeah, also a lot of questions. Don't scratch the surface ever on anything because you might not like what you find. Yeah, well, thank you very much that I could come and chat to you all about something that is understudied and probably something that there's a huge scope for within ecology. And it's Australia, all our marine species are quite understudied compared to other hemispheres, considering the beautiful diversity we've got around here. So it doesn't always have to be marine mammals. You've got a lot of cool marine stuff out here. Early embryonic death and things are not purely vertebrates. You could get it in cuttlefish or lots of other things. So there's a huge scope to understand reproductive success and failure in wildlife. But yeah, thank you very much that I could come here today.